Okay, thank you very much. So we are beginning, and it's good to be on time because every conference always says that we are running behind schedule, and so please, we don't want to do that. So with this, I invite Professor Muktish Acharya to chair the first plenary lecture by Professor Abhishek Chaudhuri, and it's on dynamics of a passive droplet in an active fluid. Muktish, you may please introduce the speaker.
Thank you, and welcome you all to the conference CMMSSP. We are going to start our first plenary talk by Dr. Abhishek Chaudhuri. Dr. Chaudhuri obtained PhD from S.N. Bose National Center for Basic Science, Kolkata in 2007. And then he moved to RRI, Raman Research Institute for postdoctoral research. And after that, he moved to University of Sheffield, University of Oxford, UK for further postdoctoral research. And he joined IISER Mohali as an assistant professor in 2012. Currently, he is holding the position of associate professor there. And his research interest is on soft condensed matter physics, both equilibrium and non-equilibrium properties, active matter, biological system, etc. So let me request Professor Chaudhuri to start his lecture.
Okay, thank you, Muktiza, and thanks to all the young organizers for this wonderful conference. And it's a pleasure to be here in one of the great bastions of India's scientific and cultural renaissance. And so I'm really delighted to be presenting a talk at Presidency. Today, of course, what I'm going to talk to you about is the dynamics of a passive droplet in an active fluid. This is already quite a lot of stuff which is being said here. So we have to go slow and see what we can understand about each of these terms. So in the last a decade or two, there has been a large amount of work on this particular area, which is called active matter. Now, what is active matter? Of course, we are not claiming here that this is some other state of matter or anything like that. What it essentially means is that the constituents or the particles or the agents, what they do is that they consume energy and perform directed movement. Okay? They could also there could be forces and shape deformations, and they could also proliferate and annihilate. So these are the things that these particles, which are these constituent particles of this particular system, can do. And it is natural that it's clear that almost everything of living systems that you will look around us would be classified as active matter. So therefore, living systems are the paradigmatic examples of active matter. Now, active matter systems are out of equilibrium. So I'll be moving away and off the mic. Is that okay? I mean, can everybody hear me at the back? Okay, so I'm fairly loud. Yeah, I generally speaking to students which are in a bigger hall, so I'm hoping that this will be fine. Okay, so what we are talking about is our active matter systems which are out of equilibrium. Now, when we talk about systems out of equilibrium, we have to distinguish the out of equilibrium systems such as this from systems where, let's say, there is an external drive. For instance, you can shear a fluid and this fluid is going to flow, but this is an externally applied stress that you are doing. And that type of systems which are thrown out of equilibrium is to be separated from active matter systems which are out of equilibrium, because here it is inherent, because each of these constituent particles themselves are, in a sense, they are consuming and consuming energy, I mean, and doing directed movement, and so they are inherently out of equilibrium. Okay? Very different from externally driven systems. Now, there are several examples of active matter systems, and here is one of the pictures of the kinds of active matter systems, especially collective behavior. What we are looking at here are you can look at on the ground or in the sky or inside the water. For instance, you can look at flock of birds or fish schools or herds, or you have human crowds. All of these collective behavior of these systems would be classified as as active matter systems. Okay, and here is a small list of reviews on this active matter physics. And as you can see, there are a lot of pioneering Indian contributions in this area, which is evident from the names which are there. Okay, so having talked to you about the examples of active matter, let us now focus on a few examples and across length scales. Okay, so I'm saying that this active matter can span length scales. So I can look at the level of the organism. So for instance, I can look at the formation of trails by ants, where the ants are leaving a pheromone or leaving a chemical cue for the others to follow. This chemical cue can also evaporate. And so there is an interesting dynamics there. And to figure out how this trail is formed, I can look at another example of how this, at the level of the organism, is looking at the flocking of birds. Now, this is something that all of us have seen around us, as is the example of ants. And so you can see this beautiful flocking together, and this entire thing behaves as if like one single organism to protect itself from prey. Okay, so you have from predators. Sorry. So you will see that some of these predators are trying to come towards it, and it is behaving as if it's like one giant animal. And how does this entire body, together behave like? And how does this flocking emerge in the first place? So these are questions that you immediately think about when you look at such systems. Now I can go take the things and move below. I mean, the length scales. I can go down the length scale. And I can go to the level of, let's say, a bacteria. And what you find here is a large concentration of E. coli bacteria. And I'll show this movie later on once again, because this is something which we'll discuss in today's talk. Is that you will see this, formations of these very, vortex-like structures that you will see here. And this is called bacterial turbulence, although this is at low Reynolds number regimes. Okay. So typically you would not classify them as turbulent behavior, because turbulence, we think about turbulence when we talk about inertial turbulence, Kolmogorov turbulence. What you see here is just because of the structure of what this fluid suspended, of this bacterial particle suspended in this fluid looks like, is what we will call this chaotic nature of this, is what classifies this as a bacterial turbulence. Well, there are different systems which we can look at. This is one of the systems that you are seeing here, but we'll come to that. I mean, if there is a, but it's not necessary. I mean, so yeah. So the length scale, you mean this length scale? I have forgotten. It's should be micron scales. Yeah. So this scale should be a micron. So. Okay. What we can also have is I can go even further, length scales even further below, and I can go to the level of one single cell. Okay. Now this is what is happening here is what is marked what you're seeing here is a cell, and what you see, these green dots are organelles which are inside the cell and are moving along what are called these filamentous tracts, which are either actin or microtubule filaments. We have the cell consists of several filamentous filaments such as actin and microtubules, and these are organelles moving on these filamentous tracts. Now, these organelles which are moving are actually being carried by certain molecular machines which are called motor proteins. Now, motor proteins, typically what they do is to take, think about them as, trains which are carrying cargo. Okay, so that's exactly what they do. They're actually moving with the organelles on top of these railway tracks. In this case, these are the filaments. Okay. Yes, yes. So, but in this case, of course, you're looking at scales further, even smaller. Okay. Now, one interesting thing that I can do is the following. So, supposing I take this last example, and now what I have is, as I said, that these are motor proteins which are moving on these filamentous tracks, and the motor proteins are carrying the cargo or the organelle. Now, think about the following. You just invert the problem. What do I mean by invert the problem? What you do is you put supposing you were able to do this experiment, you take the motor proteins, and these motor proteins, imagine that they were carrying this cargo, and you flip it and stick the head on the substrate and allow the legs to dangle. Think about them as if they had legs, similar to what it actually happens. But now, and then you put the filaments on which they were walking, put the filaments on top. Okay? So these filaments can float on this bed of motor proteins. Okay? If it sounds improbable, then let me show you that it actually can happen. Okay? So what you have here, as I said, are these motor proteins which are they're stuck at one point and their legs are dangling, and on top of that you have these filaments. Now, motor proteins themselves are of course not active. What you do is there's an ATP hydrolysis mechanism. So if you have if you provide them chemical fuel, then this can be transformed into mechanical work, and so then they will become active. So if you supply them with the corresponding ATP or GTP, then what you will find is that these molecular motors will start moving on these filaments. Now the filaments are on the top. Okay, so but the motor proteins can still walk on those filaments. As the motor proteins walk on the filaments, naturally the filament which is free will start moving on the opposite direction. And what you are going to see is from the top. Okay, now you're taking an image from the top. And so what do you see? So what you see are, if I did not tell you what these experiments were, you would not be able to differentiate them from the behavior that I showed you earlier for the flock of birds. It exactly looks like a swarming behavior or flock of birds would behave. And this is the kind of behavior that you see for these filaments. Okay. Now, I can do the same experiment, except that I will take a different set of filaments. The filaments that I have used here are called active filaments. They are what are called semi-flexible filaments. Okay. There's some bending rigidity of these filaments, which you can characterize. There's a cost of bending the filament. But supposing you take filaments which have a very high cost of bending, okay, they are like rigid rods. So you actually have those kind of filaments inside the cell as well. These are called microtubule filaments. So if you take those kinds of filaments and you perform the same experiment, microtubules, again, the corresponding motor proteins stuck on the surface of this cover slip, and then you put the corresponding microtubules on top, what you see is the following. You see that these microtubules are of course moving, but they cannot, unlike the swarm kind of behavior that you see here, what they do here is to move into these kinds of vortices. So you'll see that they will move around, and if I play the movie once again, you'll see them that they are moving around, and this moving around is happening both clockwise and anti-clockwise. And then they will form this lattice of vortices, or a vortex lattice. Yes, it emerges. This is an emergent phenomenon, of course. It was not there. To begin with, the microtubules are all in some random, they were oriented because you have just put them microtubules on there. But now what you have done is made the motor proteins active. Activity drives the motor proteins to walk on the filaments. The filaments in turn will move in the opposite direction. The filaments now is going to interact amongst each other because there's an extruded volume interaction that is going to play around. And also they are rigid, so they will not be able to bend into smaller structures. So what you see is that these are the structures, or these are the shapes that they will form. Okay? So I hope that I have convinced you that exploring active matter systems is both fun and challenging. Challenging, of course, because you see a diverse kind of set. I mean, there is no one system that you have looked at where you can say, oh, this is a common feature that you have been able to establish. And so how do you answer questions which are, which looks or seems like they are centric, they are very much, depends upon the constituent particles. But it is also fun because you see a whole host of extremely interesting phenomena. So how do I understand these systems? Well, we can class these systems. So one of the first things that you do is to class them into different kinds. And what we know now is that we can class systems in terms of dry active matter and wet active matter. So what do I call as dry active matter? This would be like bacteria once, at low concentrations, bacteria gliding on a rigid surface. Remember, I'm looking at low concentrations where I can think about particle interactions and all that. I'm not looking at a large concentration where you will have to go over to a field description and also have the fluid in it, which will put it into the class of wet active matter. So, or you can think about animal herds on land, or in the artificial realm, you can think about vibrated granular particles on a plate. This also actually can be shown to be an example of active matter. Now, what happens here is that momentum is damped by friction with the substrate. And the only conserved quantity in this case is the number of particles. So what you can do, of course, is if you look at it in a coarse grain sense, you're doing a coarse graining, averaging out, and what you can think about is that you're looking at the local density of particles. That's the conserved variable that you're going to consider. Okay. And next is that even in dry active matter systems, and this will be true even for wet active matter systems, what you find is there are several types of these particles. Okay? Now, particles could be polar. Polar meaning, remember in the beginning I was talking about the fact that each of these constituent particles consume energy and perform directed motion, which means that there is some direction that they take. Okay? So these are what I will call as polar particles. Okay? Now, I could have activity, but I could still have apolar particles. I could have a situation where activity is at both ends. Okay, think about if in one vein you can think about as if you did not know that there were these motor proteins which were carrying this filament, but you see that the filament can move in either whatsoever direction, and you think about this entire thing as if this entire motor protein plus filament object as something like an apolar system. Apolar active system, because it can move in either direction, but it has it has an active element to it because there is something which is driving it internally. So so now we have these set of polar or self-propelled units, which if I have the polar particles, and these polar particles will interact with one another, and they will tend to align in one direction. And so this is what is the familiar example of the classic Vichek model, which I will briefly talk about. And you can also have what are called apolar particles. As I said, again, this is a repetition of what I just said. So let me just show you what I mean by that, because there's a picture which is probably says better than 1,000 words. So I have a polar active particle which is of this kind. I could have apolar active particles where the propulsion can be in either direction in this way. Or I could have self-propelled rods. There's a difference between the polar active particles and self-propelled rods. Or you can, but I'm not going into the nitty-gritty details about that. Now, if I have either polar active particles or self-propelled rods, I can have two types of ordering in the system. What do I mean by that? I can have a polar ordering where all these particles are lined up with their heads in one direction. Okay? Or I could have a nematic ordering. What do I mean by that? Here, although the particles are polar or self-propelled, you see that if I coarse grain over this scale, okay, then they cancel each other. Okay? So on the average, this region seems like it doesn't have any preferential direction as opposed to this one. So this is what is called a nematic ordering of polar particles. Okay. To do this, for the ordering to happen, the ordering, of course, whether the system will give rise to a polar order or a nematic order will depend upon a lot of factors. So it will depend upon, for instance, the density of the system that you are looking at. It can be changed by changing the other external factors like temperature and stuff like that. But what I am trying to claim here is that if I have a system of such particles, then these orderings are possible. You can have situations where this polar order will emerge. You can have situations where nematic order will emerge. So that's what. Yeah, I mean, there are several examples of how these things comes about, but generally these are the kind of things that you're going to see. Yeah. So as I said, told you, when I'm looking at dry active matter, the only conserved quantity that I've talked about is the number of particles, which in this case is the local density. That's the only thing that I can look at. When I'm going to talk about wet active matter, you will see that there are lots of these. What are the conserved quantities would be? I'll come to that because I'll be discussing wet active matter in a short while. Yeah, we'll see what comes out. But in this case, this is what it is. Okay. So you can, in fact, of course, write down a corresponding. If I'm looking at nematic ordering, then I can write things in terms of what is called a nematic order parameter, which is a nematic tensor. Okay, and I can write down a corresponding equation, a dynamical equation in terms of that Q tensor. Okay, and find out the relaxation dynamics of the Q tensor. That is also possible. I mean, people have done that in dry active matter systems as well. Yes. Yes, absolutely. Yeah. So that's. So as I said, yeah. So basically, of course, people have looked at all of these things. You are absolutely right that you can think about how things can move from an isotropic state to a polar ordered state to a nematic ordering and all that stuff. So you can do find out linear stability. Here, remember that the fun part here, as I said, is the fact that activity itself is driving the systems to go into that. So they are self-propelled and self-driven. Okay? And then the interactions, and then the excluded volume interactions between them are giving rise to this kind of order. Okay? So these are all living matter. So this is like a pictorial representation of things that you have you have some, you can think about bacteria, for instance, or you can think about those examples like an ant and think that ant is like that particle here. Yeah, so it's a physicist way of looking, trying to do this kind of systems. So what can be? Clocking, of course, yes, is true. And in fact, the example is, of course, the Vichek model, where what you coming to that, like one of the examples of dry active matter is a system where what you have are these particles, and these particles are, have an orientation. So these particles have a particular orientation, and what you can do is, I mean, now if what is the next orientation that they are going to take will depend upon an average over the neighboring particles. So what you do is that you have all these particles which are pointing in whatsoever direction. And this particle will move in the average direction of all these particles within this region. So you take a neighboring region, find out all the orientations of all the other particles, and take the average and move the particle in that direction, in the average direction. So this is the simplest rule that you can think about. There are no excluded volume interactions between particles. If you do that, then what you find is the following, that at high enough densities, you will see a flocking transition of this kind. Okay, so you will see that these are the particles which are moving in this way, okay? And then at large densities, you see this flocks emerging. So of course, you can do statistical mechanics of this and ask the question, okay, if I have such a transition, can I study it? What kind of order of the transition is? Look at it with respect to density. There's something called noise in the system, which also plays a very major role. So I can play around with it and find out all sorts of stuff that we know of from statistical mechanics. Ask about critical exponents and all that. Yeah. Yes. But here there is nothing like that. This particular agent-based model is the simplest rule-based model which can give rise to a flocking transition. You don't have anything. You don't even have excluded volume interactions between particles. You just have this kind of an equation of motion, and this is enough to give you flocking. Okay? So that's like, we try to come up with the simplest examples which can give that. Yeah. So this is one example where it does so. Okay? Of course, then people have looked at more realistic systems. They have actually, tracked birds and the flocking and trying to find out how the interactions, because there's a vision cone as well, which becomes important, how they're looking at it. So all these questions have been, looked at and addressed. But those are more complicated stuff. And naturally, as the field evolved, people asked more hard questions and came up with better answers. What will be the what? Yeah. So it will, of course, once it goes into this kind of a stage, there, of course, you have looking at clusters forming, right? Yeah. So, what I'm saying is the fact that the movement of the direction in which it will move will depend upon the averaging in a local region. In fact, people have looked at what happens if you change the radius of that averaging. Like, this distance, if I change that, what is going to happen? It's R naught. Okay, and related questions. Okay, so sorry, I'm just going back. Should be moving forward, and I've already wasted a lot of time. Okay, so we'll quickly now go to what is the the point of this talk, which is about wet active matter. Now, as the name suggests, when I'm going to talk about wet active matter, then obviously solvent-mediated hydrodynamic interactions are important, which means that the particles are suspended in a fluid, and so the dynamics of the suspending fluid must be incorporated into the model. Okay, we cannot not do without that. We have to understand what the dynamics of the fluid itself is. So one must develop a description of the suspension of active particles and the fluid with a conserved total momentum. So active constituents are going to exchange momentum amongst themselves and with the environment. Examples, of course, are examples which I have already shown before. Bacteria, algae, and cytoskeletal suspensions fall in this class. I will show you an example of cytoskeletal suspension in a later slide. So what are the kind of interesting behaviors that you see in wet active matter, which are seen? So you see spontaneous flows. So we have, remember, there's an energy injection because these particles that are composed, the suspended particles are active. So there's an energy injection due to consumption of the internal or chemical energy, and this can set up self-sustained flows. Okay, so this is definitely something which is different from what you have in passive systems, passive fluids. Okay. You could also see in certain experimental realizations, you have seen what is behavior, that these rheological properties of active fluids show the emergence of a superfluid phase. Okay. Another possibility. And the third is, of course, something which I had already shown, that bacterial turbulence, remember the movie that I had shown earlier, what you find is that they develop these kind of vortex structures and resemble the hydrodynamic Kolmogorov turbulence that we know of, the inertial turbulence that we know of. Okay? So these are some of the features or some of the aspects that have emerged from in wet active matter, where the interactions between the suspended particles and the fluid becomes extremely important. So rheological. So you have flows and you're doing a shear or things like that. How do you look at the behavior? So I'll talk about one of the rheological properties. Essentially, so one of the basic concepts of microrheology is exactly what the theme of the talk is, which is basically if you put tracer particles in complex fluids and look at the behavior of those tracer particles, the dynamics of the tracer particles is going to tell you about the nature of that complex fluid. Okay? The fluctuating dynamics of these particles are going to tell you about the medium in which it is, and so that will tell you about the complex fluid itself. So this is the essence, one of the key aspects of microrheology. Normal fluids as well, absolutely, yes. I was just talking about complex fluids because in my mind, my fluids are very, very complex. So yeah, no, it's just that, yeah, you're absolutely right, yeah. Yes, but you can see it at normal temperatures. I mean, the behavior is because of that's one of the key reasons why I put it out as a feature which emerges in this systems here. But it's like none of these things are, I mean, like again, turbulent like, not really turbulent in the actual notion, superfluid like behavior. So in features, in kind of features that you see are that the system behaves like a superfluid. So. Yes. So in fact, you will, in some cases, you can actually see that even in wet active matter, you have systems where it shows negative viscosity. Now, whatever that is. Okay. Yeah. Yeah. So, can I move ahead because I have Okay, good. So building a theory of wet active matter, obviously, as you can imagine, this is not a non-trivial task because so you have density of active constituents can be very high, and the hydrodynamics feedback between the swimmers and the flow is fundamental to catch the essential features of active fluids, right? I mean, you have to incorporate all of that stuff, and the task, as you can imagine, is extremely difficult. It's not a simple thing because you have. So far, I was talking about particle interactions and how does that can be. Now you have to think about the fluid itself and how does the fluid and the particle interact with each other. So how do you go about building it? So again, the kind of class of systems that I am going to, or the kind of systems that I am really looking at are what are called pneumatic fluids. What it means is that, again, the particles which are suspended in the fluid, okay, this entire system, this is what the pneumatic fluid is. So if you think about these rod-like active particles, okay, and they are in that volume, they are suspended in a fluid. But now if I think about the orientation of the particle being given by ui, then we can define a mean local orientation. That is not so difficult to understand. So I can, in a coarse-grained region, I will be able to do that. Now, this is again something which I said before, that the mean polarization could be 0, depending upon how these rods or these particles, these polar particles are going to align. So this can give rise to what is called a pneumatic ordering. And pneumatic ordering can also come from apolar particles. So if I think about the pneumatics, now pneumatic, I am not being able to distinguish between the head and the tail. Okay. So what is the kind of order parameter which is going to give me an understanding about pneumatics? You cannot define it with a vector. What you have to use is a second rank tensor of this kind. And even just think about it as some order parameter, and it's actually what it does is it can remember that if I just have a passive system of these rigid rods, okay, which are like rods without any direction, so these are pneumatic particles, then what you can show is that it will go from an isotropic to a pneumatic transition. So isotropic means they will be all around, or pneumatic will be aligned in particular direction. This direction can be anything. Okay, these directions are all equivalent because this or that is all going to be equivalent. But so this isotropic to pneumatic transition as a function of temperature, this can be studied in terms of this tensor order parameter. So you can write down a corresponding, the usual thing that you do when you're looking at phase transitions, and you write down a free energy. And so this free energy, you expand it in terms of the order parameter. In this case, the order parameter turns out to be this Q tensor. Okay, this Q tensor is a traceless and symmetric tensor, so that definitely helps. And it measures the local degree of mutual alignment of the axis of the constituent particles. And as I said, it doesn't distinguish the head from the tail. That's what it is. Okay. So therefore, if I want to describe a pneumatic active fluid, then I have to think about the fact that there is a velocity field corresponding to the fluid itself, and there is a field which is coming from this tensor order parameter, which is a Q field. Okay. Now, how do I model active stress? Okay. So now, before I do that, just a brief, quick recap of what you see in fluids. Just called it. It would have been easier to do it on a board, because unfortunately, yeah. So the point is that, let's say that I'm talking about a fluid. Now the best way to describe a fluid is to take a small volume element, okay, which is of course larger than the tiny constituent particles of molecules, but smaller than the entire volume of the fluid itself. So you have a volume element, and what you do is within that volume element you can have several number of particles, and you do an averaging over that volume element. So what you have are essentially local descriptions of quantities like the density, you have the velocity field, you can have the temperature, local temperature in that within that volume element, and so on. So these are all coarse grained, and what you can do now is to write down corresponding. You can write down conservation laws. The first basic conservation law that you write down, and I'm sure all of you have seen this, is what is called the equation of continuity. You have seen equation of continuity, may not be in the fluid, but you have seen it elsewhere. So charge conservation, of course, is the most famous example. So now I have equation of continuity, and so I can write down del rho del T plus divergence of J is equal to 0. J is the current, and this J here, of course, the current density will be given by rho times the fluid density, right? The fluid velocity. Sorry. So rho u is going to give you the current, and now you can put it back here, and this is the equation of continuity. One interesting thing that happens is if I think about rho as being constant, okay, then this is the condition that emerges, which is that the divergence of the velocity field is 0, which is the condition of what is called an incompressible fluid. Why is this an incompressible fluid? It has to do with something with, you can write down the time evolution of the Jacobian of this transformation, of the volume transformation, but I cannot go into that details at the moment. So I'll just to say that it is divergence of u equal to 0 defines you what is called an incompressible fluid. Now what I can do is, I can also write down an equation of motion. So I'll have there's a continuity equation, and now I can write it down, the corresponding one for the momentum. And the momentum equation is of the following kind. So here is a little bit of a, so one thing is very clear, rho times A is like your force, force density actually. And so on the other side, on the right hand side, what you have is, of course, there could be several forces acting on the fluid. You could have body forces, like for instance gravity, or you could have other kind of forces coming from the fact that there are these what are called viscous stresses in the fluid. So if there's a viscosity of the fluid. Okay, thank you. And so what you can do is to write down this equation, which is rho times, this is the axis of the body forces, and you can drop the term in the absence of that. In what I'm going to talk about, I'll drop this, and I'll think about only the divergence of a stress. Now, stress is actually a tensor, force per unit area. Force is a vector, area is a vector. The it's a crude way of looking at it, but it's just saying that vector divided by vector, I don't know what to do. I'll call it a tensor. Okay, so it's a tensor. So this is what it is. So stress is a tensor, and the divergence of this stress is, of course, going to give you force. It's actually, if you look, grad gives you 1 by length, that gives you a force per unit volume. That's exactly what I should have, because on the other side I have that. It's a force density, force per unit volume. So that's fine. So this is what the equation of motion is. The interesting part, of course, here is that this capital ddt is not your usual form. This is the following form, which is a del del t plus a u dot grad. Okay, things get advected with the fluid. So that's what this thing does. Okay, material gets advected with the fluid. So now I can put it back here. Use an expression for this stress. This stress, as I said, comes from, one thing is an isotropic stress. Isotropic stress is something which everybody is familiar with, which is basically the pressure. If you think about isotropically, at all sides is equal. It's the pressure that we have, age-old thing that we have seen all the time. And the other part of it is actually what makes it very interesting. It gives you all these kinds of other terms in the equation. The first part is clear, that the pressure is the pressure part, the isotropic part is going to just give you a gradient of the pressure. The other terms are what is going to give you viscous stresses, as I mentioned before. Okay, so the divergence of those viscous stresses, and in the presence of divergence of u being equal to the incompressible fluid, then this term actually drops off, and this is the equation that you get, which is the famous Navier-Stokes equation. Okay, or Navier-Stokes equation for an incompressible fluid. Okay. Now, unfortunately, I cannot go beyond it, or I cannot go into it much deeper than this. So think about this as the classic example of a passive fluid. Okay, this is a passive fluid. There's no activity element in it at all. It's just what you see around us, water flowing and all that. Okay. So now let me just go back. I have to be quick because I'm already running short of time. Nowhere close to. Okay. So now, so that is what that part is. Now remember that in that expression or this equation that we had, I had a divergence of a stress. And now this divergence of this stress is, obviously think about it, if the normal fluid had the divergence of stress on the right hand side, that stress term would have to be changed now in the presence of these particles which are inside that fluid. These particles are active particles which are suspended in that fluid. This has to be doing something to the fluid because there's an interaction. It is changing the fluid or doing something to the fluid. How do I account for that? Now to account for that, so the first basic thing to say is maybe it is changing the stress. Maybe there's some active component to the stress. How do I calculate that? So think about a collection of swimmers, like these pneumatic particles, active forces acting on both sides. So think about for a collection of swimmers, what you can do is to think at some approximation, talk about the details later if you want to ask, is that you can think about them as force dipoles. Okay. Nice to have a force dipole. And if you can think about them as force dipole, and with, what you do here is basically, it is symmetrically separated into from about the origin, which is somewhere there, which is B here, B this side, By to that side. This B should be coming here. It's not within the brackets or outside it. So you can write down the corresponding active force or active force density in this following form. Okay. I hope this is clear because this is like a Ri plus B/2 will define this part, and Ri minus B/2 mu I will give you this one. The plus mu I and minus mu I are basically the orientation directions at the two endpoints. Okay? And so what you do is you write down the corresponding force term and do a Taylor expansion. If you do a Taylor expansion, what you see as the first order expression that comes about is this mu I, which is exactly what you saw in this tensor component. Okay? So what you can do actually is, if you do the math, the force, it's a force dipole, and the force dipole, you are assuming that these active particles are like permanent force dipoles. They are behaving as force dipoles. And if I think about that as a force dipole, I am trying to calculate what the stress component, active stress component, whether I can actually, rather than saying what the stress component, I would say, can I find out an active stress component from there? And the answer to that is yes. What you can do is, now that I have done this analysis and done the Taylor expansion, you see that this comes out beautifully as a divergence of some quantity. And that divergence of that quantity is exactly is proportional to the Q tensor that I had written down earlier. And that's exactly what it should be, because the Q tensor is basically the isotropy, is the order parameter for the pneumatic system. And in this case, it's a pneumatic, and that's how it comes out. Okay? So this is what is called, what I will call as the active contribution to that stress. Okay? I'll just, quickly go about. So yeah, so things are becoming more and more difficult. What I have is, you have an incompressible Navier-Stokes, which is given by divergence of U equal to 0. And this equation, which I have already written down or shown to you before. On the right-hand side, I have forgotten about the body forces. All that I have is a divergence of a stress tensor. That stress tensor presumably will contain all the other stresses that are there. But that cannot be only it, because there has to be an evolution of the Q field as well. Remember, there are two fields here. I have a velocity field, which is the field of the fluid in which the particles are suspended. But the Q, the Q, which is or the pneumatic themselves have a field of their own, which is a Q field. And so I can write down an evolution equation of the Q field. The first term is pretty straightforward because it gives you the advection of the Q field, right? This is This part is the advection part, and it's obviously going to be advected by the fluid. The remaining parts are, terms which comes about because once you do a very thorough analysis of it, all of these S and H turns out to be, can be written down as complicated expansions in terms of Q. I'm not getting into it. We can talk about that later, and I will show you because of, as I said. So these are, this H is actually a molecular field which comes about by taking the derivative with respect to the free energy, and this is the free energy I was telling you about, the free energy corresponding, which gives you the isotropic to pneumatic phase transition. So this has all the corresponding terms, and you can. So bottom line, I will solve for the following equations. I will have this equation for the fluid, this equation for the Q, add on to the fact that the viscous, the total stress tensor will have a viscous part. There will be an active part, which I have already talked about, and there will also be an elastic part. And this elastic part, of course, is because these pneumatic liquid crystals that I'm talking about, which are suspended in that fluid, they have an elastic Frank free energy because there is a cost in doing things like bend or splay or twist. All these kinds of deformations will bring their own elastic energy costs. And therefore, I will have to have the stress tensor will, in fact, incorporate all of those terms. What will happen to the, if you need, can you have higher order terms? That's what your question is. This is, I mean, it's obviously how long, up to what. No, this is not a thing. So this is, of course, expansion. Yes, of course, you write it down in terms of all the orders of Q. But where you have to stop is already if you get the isotropic pneumatic transition, as I said, this is, remember, a phenomenologically written down free energy, right? It's not a microscopic. You can, in fact, come from a more microscopic theory and see what terms are going to be important or not. But yeah, this is sufficient. Yeah, you want to make my life more complicated than this already is, because of sure. No, I'm just saying that you already see that we have a description of an active fluid, and it's a humongous task because you have to put all of that and numerically solve it. There is no way you can do anything else. Okay, now you can do that if you have a very good person who can do it, and this is work done by my postdoc Chamkor Singh. It took him several years to do it, almost 3.5 years to finally. But the good thing is we were able to do it because it is not only about the equations. Very few groups in the world have been able to solve those equations numerically and get beautiful features. But the question here was whether it is possible to put a passive droplet in that active fluid. Because this comes back to the microrheology experiment that I was talking about. Remember that our goal is to study systems like these bacterial dynamics, which I have already shown you, which shows, again, active turbulent-like behavior or turbulent-like behavior. And you can also have motor microtubule mixtures of some kind, which I talked about when I talked about the in vitro motility assay, where I said that I stick one of the ends of the motor to that. If I allowed the motors to also move around and not stuck them on the substrate, and the motors together with the molecular and together with the filaments were moving around at high concentrations, then you will see this kind of systems. Okay? This is what you see. So now, remember, the goal was to do a microrheology experiment, a theoretical experiment, if there is any. So how to do that? In 1999, so it's about 2 decades back, okay, there was experiments where passive tracers were put in a passive tracers put in bacterial fluid bath. What Wu and Lipscheber, famous experimentalist Lipscheber, what they showed, and these are the tracks of these 10 micron side beads in that bacterial fluid. What they were able to show is that if I look at the mean square displacement as a function of time, then it shows a ballistic behavior, so it goes as t squared, and then subsequently to a diffusive behavior. And this ballistic to diffusive transition, okay, mean square displacement, I hope most of you are aware of. So if I do that and I calculate if I look at it, then what they found was that this transition actually depends upon the bacterial concentration as well, at what concentration they are. So anyway, so this was so many years back. A recent experiment is one where they took this suspension of these fluids and these filaments and motor proteins and put these kinds of chiral gears inside it. And these are the kind of behaviors that they show. So this is a PRL in 2023, and you see these kinds of, this gear is going to rotate. Okay? There's a unidirectional rotational movement, and naturally because there's a chirality in the gear that they have chosen. So this is active fluid turn spin wheels. This became one of the physics focus articles in 2023. So the question that we now ask is, what happens if I think about not rigid boundaries, but deformable ones? Okay. So it's one, yeah. So this is making even more difficult. Yeah, that's right. So droplet in an active fluid, and that's what we have done. So just to show you, this is a velocity field. What you are seeing here, this is the movie of that droplet, and you can see this droplet is actually undulating. The shape of the droplet is undulating as it is moving in the fluid. The background fluid is the velocity. What you're seeing here is a plot of the velocity. Yes, velocity field. So it's actually divided by a root mean squared just to scale it down. Okay? But yeah, this is a velocity field. What you're seeing here is a velocity field. So sorry. I mean, I'm sorry about the bad quality of that movie. Here is a droplet. The reason the droplet, remember the droplet is a passive droplet. What differentiate between the outside and the inside is that active stress term, which was minus zeta q hat. Zeta is non-zero outside and zero inside. So it's still a pneumatic droplet. So the droplet is a pneumatic droplet in an active pneumatic fluid. Okay? So obviously, as you can imagine, there is what makes things extremely complicated is the interfacial stress terms that is going to be there as well on top of what you had before. Okay, and I'm not going into that. I'm just going to tell you the results quickly. So what you see is the following. So you can look at the droplet in an active fluid, and you can mark the trajectories of these droplets. And what you see is that if I have a larger size droplet, all of these things are scaled, so they are all in dimensionless units. So the larger size droplets are more or less localized. They don't really move about. If I look at the smaller droplets actually move around quite a bit. The smaller droplets also shows these so-called, there will be regions where they will just keep on moving around in the same region. So these are what you are seeing here is a time evolution of this. So you are looking at the interface, okay, the outside of the droplet, and as it moves in time, and you see that there are situations where it will turn. Okay, there are things where it will just turn completely. And these are the motions. And so if I look at the trajectory, what you find is that there are periods of what are called runs and stays. So it will run for a while, stay for a while, run for a while, stay for a while, and so on. If I look at the larger droplets of size which is much larger, then they will predominantly not do that kind of a motion. This is a deformable droplet. The velocity field, in what way? The question is, what do you mean by that? So if it is creating any wake pattern, no, it is not creating any wake. Which rigid one? Oh, acha, this one. This. So it very slight, but that we don't know. Yeah, we don't know whether this is coming because of the fact that these are rigid boundaries, of course. What we have here is the deformable boundaries. So the stress is actually much more relaxed around it. So in this system, the one we have, the stress is much more relaxed. Okay, because, so open questions actually. I can't tell you for sure what these systems are, but we have been able to characterize the dynamics so far. So that's what we are. So we have, of course, droplet deformations. You can look at the aspect ratio, which is the area of the droplet and how it changes with time. And obviously, as you can imagine, that they will show oscillations. The oscillation time scale, of course, for the larger droplets, you will see this bigger time scales, and for the smaller ones, you will see them, doing very fast ones, changing the shape. You can look at the same question that Wu and Dipsheber asked for the droplets, for the tracer particles in the active fluid, and ask the question in the bacterial fluid, what happens to the mean square displacement? What we find is that for us as well, the mean square displacement actually shows an initial ballistic behavior. Ballistic, it actually has a exponent between 1 and 2. And then, of course, at large time scales, it shows a diffusive behavior. As you can see, the data here is is not very good, because the number of averages and the runtimes that you can actually do is resource limited. Okay, so we could do as much and find out what they are, because they are extremely expensive simulations. Okay, each one of them actually runs for about 1.5 months to 2 months time. Okay, so that's the scale of which this goes on. Yes, the crossover depends. Absolutely, I was thinking that you'll ask the question. The crossover depends upon the particle size. It depends upon the particle size. Remember, we have not been able to change the concentration as they did for the bacterial concentration that they could do. In our case, we have taken one bacterial concentration and one concentration for the fluid and moved ahead.
Expected to change the flow.
Yes, it is.
You haven't gone that big.
We have not gone that big. Yeah, I mean, you can see that actually, if you look at this particle, it is big, but it's like, in this 400 per 400, it is like a very, very small area. Okay, so we can, I think I've run short out of time and everything. 2 minutes. I mean, I'm just taking it to the limit. So we can look at velocity correlations, and we can look at one-time 2-point velocity correlations of the fluid bath. This is the fluid bath. You're asking the question whether the fluid bath. And as you make the point, if I look at different sizes of the particles, it doesn't seem to affect the velocity correlation of the fluid bath. But what it does is that this quantity picks out a length scale. And what we find is that which we can find out from the correlation, I get a length scale. And if my radius is much less than this length scale, then you see these droplets exhibiting those drifts, okay, which means moving around. Whereas if the R is greater than this thing, the droplets moves relatively small distances. So at least what it does definitely give from the fluid bath is that the length scale that it picks up tells us how far will so it's like, to come back to your point, Anivanda, if I have a bigger droplet, then the 2 points, it's almost like uncorrelated, because their size is such that these two, if I look at the velocity correlations, then they are uncorrelated at along the diameter points of the droplet. So, yeah, yeah, well, yeah, it's a okay. I'll just quickly wrap up by saying that we have also looked at other quantities like the velocity autocorrelation functions, which shows a decay which is dependent upon the radius, and this is the autocorrelation for the particle, and it also shows oscillations. We have also been able to show the change of surface tension. So if you make the drop of very low surface tension, then the drop actually breaks up and and mixes with the outer active path. So it just actually does so. So that is also a very nice point about the simulation, that it is actually doing the correct thing, because that is what you would have expected if you have a lower surface tension that is good. So there are very interesting questions to ask here: that how does it happen? When does it break off? At what surface tension? And how does this break off actually take place? So these are questions which we have not yet explored. It's very fresh. I'll skip the effective Langevin dynamics, because it really doesn't, at the end of the day, give much in terms of coming to a theory. Okay, so I'll conclude by saying that we have studied the stochastic dynamics of a passive pneumatic droplet, which is suspended in the active pneumatic turbulence. Okay, the transition from superdiffusive mean square displacement to normal diffusion depends on the size of the suspension and the properties of the active path. Okay, we find that the initial integral length scale associated with the 2-point velocity correlation can be used to determine whether a passive object with a particular size will remain almost positionally localized or will start experiencing a drift. The motion comes out to be far from equilibrium standard Brownian motion. Few aspects of the dynamics can be qualitatively described by a Langevin model with very few. The effective Langevin dynamics, which I skipped, doesn't do much justice to the kind of results that we have got. So anyway, it's there in the paper. So if you wanting to go back and look at it. And so I think we have, and to some extent, we have nailed the problem, if you may say so. And the acknowledgements, of course, is Shamkor, who has done all of this work. He's an inspired faculty at the Central University of India. He was a postdoc of mine, now he's an inspired faculty there. And Shrudeep Haldar, who is currently doing a PhD and is working further on this problem. And with that, I would thank you and thank you for this.
We will talk on this active matter. Now the session is open for you. But very limited number of questions we are already running.
Short of time, yes. Yes.
Yes, Anibar, please.
Yes. So if I also work from 2 to 1, yes, it does. So if I at what? So we looked at, for instance, we went to lower the sizes which are 8, which is typically a small one, not point. Point, I don't know how to, because you have to take into account the boundary, Yes, absolutely. And they have seen this.
What does deformability mean?
So deformability in this was a kind of a proof of principle kind of thing, because what we were saying was that Wu and Lipscheber have done the tracer particle thing, which showed this ballistic to diffusive crossover, and actually they made the point that the alpha it's not alpha equal to 2. It's t to the power. It doesn't go as c squared. It goes somewhere between 1 and 2, and we actually obtained that in our simulations as well. The value, and the other thing, of course, is the dependence on the size. So they, in fact, made the point that the effective diffusion coefficient will go down with the radius, which is kind of, if I think about k2 by 6 pi r, and that's the typical thing, 1 by r dependence is going to be c. And well, we do see the fact that the effective diffusion, it's very difficult to figure out the effective diffusion coefficient from these plots, but we see similar kind of features. We try to do more number of averages and go to longer time scales, which is a crazy point. But because you can imagine, if I want to get the effective diffusion coefficient, I have to fit this with a t, and that becomes a problem. So this kind of sets us the main things that are important about this deformation fluctuations. The fact that if I go to lower surface tension, this takes up and happens. And also these runs and stays that you see. See, nobody so far, in the context of active fluids and droplets in that, have talked about, people have talked about when they look at active fluids with particle-based simulations and all that stuff. This has been done. But even, and run and tumble, this is kind of saying that the active fluid itself is kind of giving some sort of a run and tumble behavior to these particles. Those are the salient features I would stress, rather than the mean square distance. The mean square distance is important to show that they were actually coinciding with experimental. It's scaled units, but
Timeline. So that from the derivation.
Yeah,
dimensional analysis.
Yeah,
If you have non-dimensional the differential equation.
yes, so the entire thing is made dimensionless. So therefore, everything that I'm talking about here is dimensionless. So because that's very crucial in this kind of things. You do not want to keep dimensions hanging around. The first thing is to just make them everything dimensionless and then work with it. So they have been done.
Takes dimensions too.
Oh, yeah. That is crazy.
Computation takes dimensions. 1 month.
No, so what I mean is that in that dimensionless times units, and if I have to run for whatever scales, I want to see a long enough behavior to be able to capture mean square displacement, for instance. Otherwise, you will not see it. You will only see the short time behavior, but you will not see the long time.
Okay, I know that you have many more questions, so I request you just talk to him in the lunch room.
Yes, yes, absolutely. Yeah.
So let us thank
absolutely.
the speaker, and as a token of appreciation, the student will give something.
Oh, Mon.
Return.
Thank you. Thank you. Wow.
Thank you, Abhishek.
Yeah, thank you.
Kolkata. So, কোনো comments করতেছে? তাই নাকি? লাগানো হয়েছে? কাজ হচ্ছে শুরু হয়েছে? চলছে? না না দিয়েছে। দেখুন দেখুন দেখে 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 লাইটটা বললে ঠিক আছে অ্যাক্সিডেন্ট আমরা চলে যাই।
Thank you, Sagnik. So this is our first oral presentation session for the day. In this session, we have 5 speakers. Now, a general notice to everyone. Every talk will be of 12 minutes duration, followed by a 3-minute quick question-answer session. Now, I shall be giving a warning after the first 10 minutes of this 12 minutes. So, let us begin with our first speaker for the day. Today, our first speaker is Ritik Sarkar. He is a senior research fellow at the Department of Physics of Complex Systems, SN Bose National Center for Basic Sciences, Kolkata. So over to you, Ritik.
Good afternoon, everyone. So first, I would like to thank the organizers for giving me opportunity to present my work in this conference. So today I'll be talking about transport properties of a harmonic chain driven by active Rubin-Bach. So before, Okay, so before discussing about what is active Rubin-Bach or like what is it I mean by transport properties, first I will just motivate showing some experimental and theoretical results. So I'm not wasting any time by introducing this active particle because Professor Chaudhuri gave an excellent introduction. So just this cartoon. So they have an active particle that takes up energy from its environment and stores it as an internal energy, and then it converts it into some kinetic energy and performs its own dynamics. So there are several examples of active particles. In the microscopic world, there is bacteria, genus beads, and in the microscopic world, there are hexbugs, birds, flock of birds, and crowds, etc. And here it is a schematic view of what I mean by active reservoir. So there is a collection of active particles, and there is some kind of interaction is there. So now why we want to study this kind of stuff? So because of this kind of these results, so this famous Wu and Lipschewer paper and Maggi, so people have reported some emergent features when they are studying the dynamics of single particles immersed in this kind of active reservoir. So they have reported about negative viscosity, modification of equipartition theorem, emergence of memory, etc. So now it is intriguing to ask what will happen if we attach an extended body to such kind of active reservoir. So just like this picture. So don't take this picture very seriously. It is just a cartoon. So imagine there is a polymer chain, and it is attached to two baths of active particles, like a bath of bacteria A and bacteria B. So in the ends, there are some kind of motion and pressure because of these active reservoirs, and we expect that there will be some energy, mechanical energy flowing through the system and all. So we want to study this kind of system. Now that is our basically model. So we named it as an active Rubin bath. This is the active version of famously studied Rubin bath, thermal Rubin bath. So this is just active particles. They are attached to its nearest neighbor by a spring. And to see the effect of this whole reservoir, we attach a passive probe particle at the boundary. So to see the effect of this active reservoir, we have to understand what will be the equation of motion of these passive beads. That's why we have to write down the equation of motion of this total m number of particles, and this is it. So this is written in the overdamp limit. So this nu is the dissipation constant, dissipation constant acting on each particle, and this FL and FL are the active forces acting on each oscillator of this reservoir. Now we have to tell something about the dynamics. So the dynamics, this dynamics of this active noise, this is taken to be one-dimensional run and tumble motion. So the velocity of each oscillator tumbles between V0 and minus V0 with a rate alpha. So the definition of the activity is 1/2 alpha. And as this is a well-known example of telegraphic process, so the noise correlation is given by this equation. And now we can solve this equation of motion, and we can substitute the Ym in this equation of motion of the probe particle. And after some algebra, we have this kind of generalized Langevin equation. So the passive probe particle is acted upon by some dissipation kernel gamma t and some effective noise sigma t. Now the nature of this dissipation kernel and effectiveness. So the dissipation kernel in time domain looks like this. This I0 and I1 are the Bessel function, and this theta t ensures that the dissipation kernel is 0 at time 0. And the long time decay is given by like this 1/√T behavior. And the dissipation kernel in the frequency domain has some real part and imaginary part. And this is the frequency dependence of this real part of dissipation kernel and imaginary part. And this dissipation kernel actually depends on the structure of this active reservoir. So actually the friction coefficient nu and the coupling strength lambda. And this next is the correlation of this effective noise. So the correlation of the effective noise in the frequency domain is given by this G tilde omega tau is known as the spectra of this active reservoir. And this spectra of this active reservoir is related to the real part of this dissipation kernel by this equation. And this equation makes why this study is interesting. So because of this relation, it is interesting to study because it is a example of modified fluctuation dissipation relation. So if fluctuation dissipation relation holds, then the ratio between the spectra of the active reservoir and the real part of this memory kernel, that will be a constant that will be named as the effective temperature of this bath. But here it is a frequency dependent part. So for a given activity, we cannot assign an effective temperature for this bath. So I will show some results comparing these two, like where the fluctuation dissipation relation actually modifies and where it is not. So now our general framework is complete, and this is what we want to study. So we have a passive oscillator chain in this between, and there is two active Rubin bath attached to two ends. So both these two active baths are having a different activity, and now as we have the understanding of memory kernel and effective noise, we can forget about the structure. We have to look on the boundary oscillators. So the boundary oscillators are subjected to two effective noise coming from this reservoir, this sigma 1 and sigma n. And both oscillators are subjected to this kind of dissipation kernel, which is same for both the boundary particles. And now the transport properties and the stationary state can be characterized in terms of correlation functions and average energy current flowing through the system. First one is equal time spatial correlation. So this is nothing but the average of velocity of Lth oscillator and its neighbor and next nearest neighbors in that sense. And in the thermodynamic limit, if we calculate this correlation function for the bulk oscillators, it is just the sum of two exponentially decaying function. So each one is coming from two active reservoir at the two ends. And here there is an emergence of a length scale. So this Li that depends on the activity of the length scale. And in here it is the simulation result. So this is the QL actually dominated by the active reservoir with higher activity. Now, in comparison to where the thermally driven bath, where the fluctuation dissipation holds, these length scale are actually absent in that case. So this VL prime is just T average by M Kronecker delta LL prime. And the next correlation function is two-time velocity correlation function. That is the correlation of the velocity of the same particle, but there is some time lag between them. And in a small time regime, for the activity-driven case, we can write this as a series in time. But this curly tau is given by this equation, and this is the small time behavior. So we are up to this quartic order, and in the large time limit, this is actually goes as this Bessel J kind of nature. This Bessel J kind of nature has a like oscillatory behavior, but that decays as 1 over root t. So it is in the simulation, it is the envelope of this function goes as 1 over root t. And so when we compare it with the thermally driven harmonic chain, this is for whole time. So for small time and the large time, this is only this governed by this Bessel J kind of behavior. And now we can talk about the amount of energy transfer between the Lth and L-1th oscillator. So this is the actually instantaneous energy current between these two oscillators. And in the thermodynamic limit, we have this kind of Landau-like formula. So this has a phonon transmission coefficient given by this omega square mod G1 n square.
2 more minutes.
Yeah. And the contribution from reservoir spectrum. And this is the nature of average energy current in the stationary state as a function of activity of the left active reservoir. So two features are there. So there is a negative differential conductivity because dj at d tau is negative in some parameter region, and this active current shows two sign reversal. So this crosses the 0 point twice. So one is when the activity of the both reservoirs are same, and another one is tau 1 star, which we name as a non-trivial sign reversal point. And these two features are actually absent in the case of thermally driven chain. So that is all about this activity driven chain. Now this is so we can summarize our results. So we proposed a model of active reservoir in terms of RTP chain, and characterize it in terms of dissipation kernel and effective noise, shows that there is a modified fluctuation dissipation relation holds between them. Then we studied the non-equilibrium steady state of this activity-driven harmonic chain, and we find that there is a finite correlation length, and the average energy current shows sign reversal, and negative differential conductivity is also reported. So this is just an advertisement. So our work is now accepted in PRSA. So there, I have like apart from these results, we also generalize it in terms of other dynamics of activity and also modifying the linear nature of active reservoir. So thank you for listening.
Thank you, Ritik. So now we are open to questions from the audience. Any questions?
Yes, sir.
Yeah.
So basically, by increase the current that goes to that.
Yes.
Is it just depending on which path is stronger and which is weaker? Does it decide the direction, or there is something more?
No, it actually if the activity is higher, then energy starts to flow, but then it shows that there is a sign reversal. So that is why it crosses the 0 point twice. So
Even if one path is more active than
Yeah.
the other,
Yes.
it's not unidirectional.
No.
It can
Yes.
depends on tau.
Yeah.
And one person and the other kernel
Yes.
you chose.
Yeah.
Is it because you could solve them analytically, or is there some other reason?
No, we can like from the structure it arises. So it is not a specific choice. Because of the structure, the memory kernel comes. So so I can show you the actual structure. So in the actual limit, this is given by this lambda mm. So this is this matrix, and if we take this to n going to infinity limit, then we get that kind of I0 I1 behavior. But it is not a choice. This is coming from the construction. How noise is affecting the dissipation? Yeah, so the noise correlation and the dissipation is related by this equation. So I've shown this relation. No, not really. There is only some relay that the noise correlation and the dissipation is related, but it is not slowing or anything like.
What is the relation? The effect of the noise. Is there any effect?
Effect means whether it will.
How it is affecting the distance? Noise. Can I say that noise is perturbation?
No, of course, noise is perturbation.
So how it is so it should affect the distance. That's one effect on
One thing is that so this dissipation and noise, the noise and the dissipation. Noise is actually dominated by the activity of the particles, right? So this is related. This is actually the noise correlation is actually proportional to the dissipation, but there is no reason to like only tau dictates the amount of noise, not the dissipation.
If there are no more questions, so let's thank the speaker. Now, our next speaker for the day is Poulomi Wag. She had completed her master's from Presidency University, and currently she is working as UGC JRF at the Department of Chemistry, Presidency University, Kolkata. So over to you, Poulomi.
Hello. We have studied how the particles diffusion gets affected when they are passing through a narrow channel because they have some sort of interaction with the channel walls. So here is a picture of the active genus type of particles which we choose for a simulation. So these particles, they have two different hemispheres possessing different properties, and one of the side is the active side which reacts with the solvent and produces some fuel which drives the particle to a particular direction. And to understand the uh, how can I play the video? These are the movies.
পিডিএফ তো আচ্ছা ওই পিপিটিটা পাঠিয়েছি আচ্ছা পিপিটি
Presentation, not this one. Presentation, second presentation.
ও সরি সেকেন্ড হ্যাঁ এটাই আচ্ছা এটা কিন্তু পিপিটি তো নেই
মেলে তো পাঠিয়েছিলাম পেন ড্রাইভ।
না এতে আছে আচ্ছা এক সেকেন্ড
There are a movie should be present to understand the dynamics of the particles. So the particles movement is basically the interplay between translation and rotational noises and self-propulsion velocity. Then again, because of the fabrication defects in this genus type of particles, they also possess some additional feature, which is the intrinsic chirality. So this chirality makes the particles to rotate in a particular direction, clockwise or anti-clockwise. So in their motion with self-propulsion velocity and diffusion noises, we can also see the rotation, clockwise or anti-clockwise rotation because of the torque. So this type of situation we want to simulate where we are putting a dilute solution of active genus kind of particles in between a narrow channel, and when the particles distance is lesser than a cutoff distance, then the particle diffusion gets affected because of the channel wall, and this will make some
পিপিটি সামথিং করাপ্টেড আই থিংক
জাস্ট মুভিটা প্লে করা যাবে না
এটা
উপরেরটা
এই যে
এটা প্লে হচ্ছে এটা
এই যে এখানে এই যে স্ক্রিন করুন ইয়ে করে দেবেন মিনিমাইজ এই দেখো এই দেখো
Oh, okay. So we can see here in a small movie that how the particles motion, how the particle is moving here in a box which has some reflective boundary, and the motion is because of the diffusional noises and self-propulsion velocity. And we consider the blue part is the active part, and the red arrow directs the motion of the particle. here also the particle has some intrinsic torque, and these are the chiral active particle, and it has some anti-clockwise rotation along with the noises and self-propulsion velocity. And we want to simulate this type of system, and why we are considering that particles have some sort of interaction with the world, because the particle have different halves with different properties, and they may interact differently when they are near the wall, and that will stabilize some specific orientations of the particle. And when the particles are near the wall, the active halves of the particle are not equally exposed to the fuel, which also makes the some specific orientations more stable than the rest. We model the dynamics of the particle by overdamped Langevin equation, where the V0 cos theta and sin theta terms are the components of self-propulsion velocity, and their Gaussian white noise is present, and the theta part is the orientation of self-propulsion velocity with respect to the channel axis, and the first term in the equation of theta is the intrinsic torque of the particle. Then we considered potential which comes from the interaction with the wall, and the noise part. So we have considered three types of configuration. In configuration 1, the self-propulsion velocity direction is perpendicular with the wall, and we have chosen this type of potential form of potential for that. And configuration 2, where the self-propulsion velocity makes an angle of 45 and 135 degree with the wall, channel wall. And third configuration, where the self-propulsion velocity orientation is parallel with the channel walls. So in the when we plot the results of the diffusion with the interaction strength of the wall for configuration 1, where the self-propulsion velocity is perpendicular with the wall, we saw this type of plots where with increasing in the strength of the interaction with the wall, the diffusion gets decreased. And if we try to explain why this is happening, because when the particle's orientation is perpendicular to the wall, then the self-propulsion velocity helps the particle to stick with the wall, and then only because of the rotational diffusion, this orientation gets fluctuated from the 90-degree angle, and the small fluctuation is denoted by delta theta, and we can relate this with a dichotomous fluctuation, and the particle is reversing its direction between two states of minus V0 sin delta theta and plus V0 sin delta theta. And we, for this type of processes, the diffusion equation becomes like this, and since the angle is small, so we can simplify this equation further, and from this we can see that the diffusion decreases with the square of the increasing alignment interaction strength. And when the interaction strength is very high, then the particles get stuck with the wall so strongly that only because of the translational diffusion, when their distance is larger than a cutoff distance and they move away from the wall, then only the reversal of the direction is possible. So in that case, the diffusion is insensitive of the wall interaction strength and only depends on the thermal rotational noise T0. That was for the chiral particles. And now if we take chiral active particles, we saw that the diffusion exponentially increases with increasing in interaction strength of the wall. Because in case of chiral active particles, because of the torque, we have considered that here the torque is moving the particle towards the left hand side, and it tilted from its stable orientation and keeps sliding along the wall with an tilting angle of theta bar. And to reverse its orientation from this direction and make it opposite, we can explain it by a arrhenius type of barrier crossing process, where the reversal time is equals to is proportional to the exponent of the interaction strength, so that this result fits well with the simulation data, and with increasing in the interaction strength, the diffusion of the particle increases. And if we take the intrinsic torque in a range where the torque is lesser than the rotational diffusion coefficient to very much higher than the rotational coefficient, then we saw the curve passes through a minima, then it reaches a maxima, and eventually a limiting value. Because when the intrinsic torque is much smaller than the noise, then the particle practically acts like an achiral particle. So in that case, we previously saw that for achiral particles, the diffusion gets decreases. And again, with increase in the chirality of the particles tilted in an angle and stick with the wall and keep sliding in a direction for a long time, which makes the diffusion transients very longer, and exponential increase of the diffusion. And further, when the strength is too high, the diffusion is insensitive of the interaction with the wall and only depends on the thermal noises. Similar result is observed in case of other configurations like configuration 2. In configuration 2, the result is quite similar as the chiral particles, and in configuration 3, when the orientation is parallel with the wall, then for chiral particles, there are two stable configurations are possible. In configuration SC2, the particle's velocity orientation is away from the wall, so this configuration has no contribution in keeping the particles near the wall. And in stable configuration 1, that is also same as the chiral particles, and we saw an exponential increase in the diffusion. So this is the summary of the work. Where for the stable configuration 1, we can see that with increasing in the strength with the wall, the diffusion gets decreases, and for chiral particles, we can see an exponential increase. And similar trend is also followed for other configurations too. I want to thank my lab members and my supervisor. Thank you.
Thank you, Bolumi. So now we are open to questions from the audience. Any questions?
Single particle genus, is there other experiments where they see this chiral behavior?
Yes.
There are examples.
Yes, I have one publication where we put some particles of opposite chirality, like a mixture of particles, in convective flow, and we separated them in the two ends of a channel.
Oh, I see. Experiments.
Not experiment. That is a simulation data.
So as
experimental papers like Steve Darney.
That's from chiral genus. If I have two, I know that they have two genus particles together that can show this behavior. But I was not aware of this. It's very difficult to do. The other question is, in this case, maybe I missed that part where the wall, the effect of the wall. I mean, the particles, you're putting the particles at the wall. Is that how you're looking at it?
Not in the middle of the channel.
In the middle of the channel. And you're allowing the particles to diffuse. So, but the wall effect is always felt in the equation, isn't that correct?
No, only we are considering the wall effect when the particles are closer to the wall.
So there is some condition which is
cut-off distance. When their distance is lesser than the cut-off distance.
Yes.
So the equation, I can actually understand the scale comparing this book, that like at the beginning, there was a cost down here, the potential and in the beginning. Three equations you showed, yeah. Second term you will write out equation.
Okay, so here the
What
second.
is the cost?
Here we have chosen a potential like of the wall like this, so we can maximize or minimize the interaction strength in our computer simulation. So first, the omega part is the omega bracket y. That part is the strength of the wall, and the cos this form is chosen how the potential, like depending upon the orientation of the particles, which potential value they have minimum energy. Okay, and the d part is the degeneracy term, like how many such orientations are possible. We also considered that. And the phi part is the phase factor.
Any more questions from the audience? Okay. If there are no more questions, let us thank the speaker. Now, our next speaker for the day is Dr. Poulumi Ghosh. She is currently an assistant professor at Institute of Health Sciences, Presidency University, Kolkata. So over to you, Dr. Ghosh.
Good afternoon, everybody. So I did my PhD from IIT Kharagpur and postdoc from Cincinnati Children's in the area of biomaterials and tissue engineering. Then I joined CSR IICB as an inspired faculty and started the work on nanofibers for use as hemostatic bandages. And after I joined Institute of Health Sciences in 2022, I'm still continuing this work, which I want to present here. So what is a hemorrhage? So any hemorrhage is the oozing of your blood due to ruptured blood vessels. So whenever there is a cut in your finger, after some time it will stop the bleeding. But if the cut is massive, for example, in the military combats or in the road accident or surgical table, then a lot of blood keeps on continuing oozing out. So here the time is a crucial factor. So for that type of systems, we need the interventions of biomaterials. Biomaterials are non-viable materials that can interact with living organisms. They can be synthetic polymers or natural polymers, and they can be formed into different shapes such as nanofibers, sponges, hydrogel, etc. One such biomaterial which is marketed by Johnson & Johnson is Surgicel, which can stop the bleeding of blood. So as I said, nanofiber is one kind of biomaterials. This is fibers in the diameter range of nano, lightweight, tunable porosity, and has high surface area to volume ratio. So electrospinning is one kind of a technique to produce nanofibers. So what happens is here is that in the syringe, we put our polymer solutions and we apply a voltage bias. Okay, and say this is in the needle we provide a positive end, we are connected to a positive end, and the collector we are connected to a negative end. So when the polymer solutions come out from the syringe, it is typically spherical in shape. But due to repulsion of the positive forces, the spherical droplets deform and form a cone-shaped structure, and ultimately it forms a jet, which is then collected in this collector. So this nanofiber formation depends on the chain entanglement of the polymer, because if there is no chain entanglement, there will not shear thinning property, proper rheological property, and this again depends on the molecular weight of the polymer, the viscosity of the polymer, and so on. So polycaprolactam is one such a polyesters that is widely used for nanofiber and biomedical applications. It is biocompatible, FDA approved material. So if you see, this polycaprolactam nanofibers has been used for nerve tissue engineering, bone regeneration, cartilage regeneration. But if you look at the polymer molecular weight, it is molecular weight is from 80,000 to 90,000. Because this such high molecular weight can lead to chain entanglements, nanofiber formations, and formation of nanofibers of 150 to around 150 and or more nanometer range. But we hypothesize that if we are using this low molecular weight polymers, then we can find even reduce the diameters of the polymers. But the literature says that if we are using low molecular weight polymers, it is true that we will get an ultra thin nanofibers, but we will get a bead on a string-like structure. So it will be not an homogeneous nanofibers. And this will lead to the drop in the mechanical properties of these nanofibers. So actually, when we actually electrospun our PCl high molecular weight, we saw the same result. For the high molecular weight, we got nanofibers, but for the low molecular weight at different concentration even, we got this bead on a string-like structure, which was undesired. So we hypothesized that the reductions in the beads and decrease in the diameter can simultaneously be achieved if we can blend some charged polymer. That can increase the chain entanglement as well as increase the conductivity of the solutions. So our objective was to synthesize or develop an ultra-thin nanofiber and assess for their rapid hemostasis, that is, its ability to stop the bleeding. One of my PhD scholars had already extracted keratin from human waste by green chemistry and reduction methods. And we also found that keratin is actually helping in hemostasis. So we started blending the keratin with PCl polymer. So if you see, when there is no keratin, the conductivity is quite low. But when we are starting adding keratin, the conductivity is increasing. But we had to maintain a ratio because if we add a lot of keratin aqueous solution, there was phase separation that we got. So we had to optimize the ratio of the PCl to keratin. And once we got some fibers of PCl keratin, then we started increasing the keratin concentration. And then we found a bead-free, defect-free morphology of the nanofibers in a particular ratio. We call this as PLK-UT. We also spun a polymer high molecular weight PCl with keratin. We named it PKH and a PCl high molecular weight nanofibers. Then we wanted to do some protein adsorption assay because in our blood there are a lot of protein, albumin, hemoglobin, fibrinogen. So we fluorescently tagged fibrinogen and saw it under a fluorescent microscope. We saw that our ultra-thin nanofibers absorb the most amount of fibers. This is not surprising because it has a ultra-thin morphology and it has high surface area to volume ratio, and so it could absorb a lot of protein. Then we did the cytocompatibility with the cells in line 29. We found that all of the nanofibers could support the cytocompatibility. Now we wanted to test that whether these fibers can help in achieving hemostasis or not. So when a wound happens, the platelet will come and adhere to the wound site. It will get activated and it will get, and in the same time, the fibrinogen from the blood, under the influence of thrombin, will form fibrin all over, and this is how blood clots are formed. So first we wanted to test whether our fibers could support platelet adhesion or not. Okay, so we found that all our nanofibers could support the platelet adhesion, but our commercially available Surgicel, gauze and bottle-clot gauze, they could not help in the platelet adhesion. Moreover, if you compare the three nanofibers, here in the ultrafine nanofiber, you can find layers of platelet one over the other. This is again because of the high surface area to volume ratio that could afford more cell adhesion. Then when we wanted to check whether our nanofibers could help in the activation of platelets. So CD62P is a marker for platelet activation. We found that all the nanofibers could equally activate the nanofibers in comparison to the control and marketed controls. So inspired by this, we wanted to develop a product. So we directly electrospun PCl high molecular weight or our ultra-thin composition onto a gauze, and we got this kind of product. Then we did some whole blood clotting kinetics. So when we had incubated human blood, into our samples, and then we put distilled water, because distilled water will go and lyse the RBC which is not in the clot. So RBC, from the lysed RBC, some hemoglobin will come out, and the absorbance we measured. So it will, because it is not in the, this hemoglobin is not in the clot, so this will give an inverse, this is inversely correlated with the size of the clot. Here you can see in our ultra thin nanomorphology and gauge, the absorbance is the lowest. So the blood clot size was the highest here. And we also did the same analysis of whole blood on our nanofibers. Here we saw an unusual thing, like all the blood clots are biconcave in nature. But our ultra-thin morphology, the blood clot, the RBC changes its morphology. It is like showing some spiny structures, and in other type, it is showing some polyhedral structure. So when we found literature, we gone through the literature, we found that this shape changes actually helps in the stopping of the bleeding of the blood. And then, inspired by this and encouraged by this, we went for an animal model testing. So, we in a liver model, we cut an incision of 1 centimeter by 0.5 centimeter, and then applied our materials onto that. So here you can see the time taken for blood clotting was the lowest for our ultra thin fibers in compared to
2 minutes.
Yes, in compared to the controls, the gauge and surgical, etc. Also, if you see the gauge and the PCL high molecular weight, they retain the blood morphology. But for our ultra-thin nanofibers, it was polyhedral. So it is a wonderful thing, because when it is becoming polyhedral, it is like forming a bridge and a wall-like structure. It is not allowing the blood volume to escape. And that is how it is stopping the bleeding of blood. We also wanted to check the biocompatibility so we to retrieve the liver section and did the HNA staining. We found no immunological response, so it was quite compatible. So in summary, when PCL low molecular weight was spun, we found lot of bead on a string-like structure, but when we added keratin, a charged polymer, we could get a nanofiber, ultra-thin nanofiber of around 50 nanometer, which was quite low. And this helped in blood adhesion, protein absorption, blood adhesion, platelet adhesion, activation of platelets, and improved hemostasis both in vitro and in vivo. So I will like to acknowledge my lab and Dr. Shubhro Dotto, a hematologist from Tata Medical Center, from whom I get the blood samples. And this entire work has been done by my PhD scholar, Shivangi, and my funding agencies, and both the infrastructure facilities of IISER as well as Presidency University. And thank you, the organizer, for allowing me to present here. Thanks.
Thank you, Dr. Ghosh. So now we are open to a couple of quick questions. Any questions from the audience? Yes.
So you talked about nanofibers, but in your calculation, always the length is micrometer. What is so?
Which one?
All the calculations you showed, you see the length is micrometer.
Oh, that is RBC is in micrometer strength. That is fine here. But if you see the actual nanofiber, so this is a nanofiber. This is actually a nanofiber sheet. I can show you the nanofiber sheet. So if you zoom it under the same microscopy, so this is a sheet of nanofibers that can be used for clotting the blood. So whenever there's clotting, you put this nanofiber sheet above. But if you look at this in the SEM, you will find that each of the strands are in the nanometer range.
The same question is that have you commercialized your product? Or yes?
No, I will like to the help of Presidency University to patent and commercialize those.
Does this platelet counts affect the blockage? I mean,
Yeah,
differs from platelet counts.
Yes.
If there is low platelet count, how does it work? Does it make any difference?
So platelets in our body are in the range of millions of cells. So whenever there is a wound, the first blood that comes for blood clotting is the other platelets. So our materials have the property of activating the platelets, like allowing it to adhere and activate, so that the blood clotting cascade can start.
I have a question that students are into or into you are person. How long the wound should be? Small, large, medium. The wound size is one.
So we have done in the in the animal model basically, and in the animal model we had done ear like incision of 1 centimeter length and diameter, like depth is 0.5 centimeter. Similarly, for the rabbit liver also we had made incisions, same dimension, and also for the rat tail. So all these are our in vivo models. Weight percent is 20 weight percent.
And what is the typical diameter, like in nanometer?
We could go up to 50 nanometer.
So this showing that at the one side, the RBC from biconjugate goes spherical, speculated. Why is that? Because of something being changed or something?
No. Actually, when the blood cells are seeing the materials, so blood cells when it's coming in contact with the material, it is seeing, like it is sensing the different things like roughnesses and contact. So that is how the platelets and RBCs has a lot of motor proteins. So because of this change in the topography, so it is also the motor proteins is also changing in their shape. Cytosol is nothing but an oxidized cellulose. Okay, and it has nothing like it just works on absorbing the volume of blood and then concentrating the clotting factors, and it has no role in activating the platelets and RBC. So it just works by concentrating, like absorbing a lot of blood, just like gauze, and it is made up of oxidized cellulose. So it came into market in 1980s, and still year after year they are just like changing a bit of the composition and then they are marketing. Yeah, that's all. Thank you.
So if there are no more questions, let us thank the speaker. So our next speaker for the day is Olivia Mullick. She's currently a research scholar at the Department of Physics, Presidency University. So over to you, Olivia.
Good afternoon, everyone. Before I start, I would like to thank the organizer for giving me this opportunity to present my work here. Today, my presentation title is Critical Behaviors of Anisotropic XY Ferromagnet in Presence of Random Fields, a Monte Carlo Study. To keep the presentation short but sufficiently self-content, first I will sketch the outline of the presentation. First, I will give the introduction of XY ferromagnet, that is the continuous symmetric magnetic model. Then I will discuss how anisotropy plays the crucial role in spin orientations and how we have introduced the quench random field into the model. I will briefly discuss about the numerical approach we have used. Then I will discuss the results followed by a summary. So XY ferromagnet exhibits interesting characteristics due to the spin can rotate in a plane which is different from the Ising model where the spins are restricted to just two states. Here the spin can point in any direction in two-dimensional plane. The Hamiltonian can be written by this equation where J is the nearest neighbor ferromagnetic interaction strength and S is the 2D unit length spin vector. One may think it is a rotor which can orient in a plane from 0 to 2 pi orientations. In the Hamiltonian, the cosine terms encapsulates the SO2 symmetry because it's invariant under rotations. BKT transitions where the unbinding of vortices disrupts this SO2 symmetry exhibits interesting phases without having long range order. This is the topological phases. Superfluid helium, thin film, superconductivity are great example of 2D XY ferromagnet. But whenever we are going to higher dimensions, we can see the long range order or conventional symmetry breaking ferro to paraphase transitions. For three-dimensional XY ferromagnet, the ferro-para phase transition temperature is equal to near about 2.206. But these were the cases for isotropic system. Let's have a twist. Instead of having absolute rotational freedom, give this spins preferred alignment directions, which in literature is termed as anisotropy. To quantitatively understand this is the Hamiltonian of the system, where omega is the anisotropy parameter. You can see that if omega is 0, this is the isotropic XY ferromagnet. If omega is equal to 1, this will map to the XX model, still retaining the continuous symmetry. Anisotropy influences how the spins collectively orient and how they respond to the external factors like temperature, magnetic field, etc. For positive value of omega, the spins prefer to be aligned along the x direction. Similarly, if we put omega as a negative, then the spins prefer to be aligned along the vertical direction. So this anisotropy breaks the inherent SO2 symmetry of the system. So quench disorder affects equilibrium phase transition in many ways. So it is natural to ask whether there is a long range order in case of anisotropic XY ferromagnet in presence of such random disorder. Here we have introduced the disorder in the form of random field, that is HI. From literature, we came to know that there is no long range order for 2D and 3D isotropic XY ferromagnet in presence of random field. The lower critical dimension for having a long range order is 3.9. Here we have introduced the random field in such a way the randomness of the field is governed by its directions only. The field angle can be varied from 0 to 2 pi uniformly, so that the average value of the random field is 0. We have employed Monte Carlo simulation technique to study the equilibrium behavior of classical XY ferromagnet. It should be mentioned that the lattice dimension is 3 and the spin dimension is 2. A simple cubic lattice of size 20 is considered and periodic boundary conditions are applied in all 3 directions. Metropolis single spin flip algorithm is used to update the spins. Here we have shown that the quantities of interest for the equilibrium phase transition. The XY ferromagnet has 2 components order parameter. The magnetization is defined by this equation and the corresponding susceptibility is given by this. We have also measured the specific heat of the system by this equation, where E is the energy density of the system calculated from the Hamiltonian. If the system is gradually cooled down from a high temperature random states, the spins align and the magnetization grows. Corresponding susceptibility also shows the peak at specific temperature. We measure the critical temperature from the position where the susceptibility gets its maximum. So here we can see that the thermodynamic observables for 2 different strength of random field, the critical, if we see the susceptibility peak, if we increase the strength of random field, the critical temperature decreases, that is the susceptibility peak shifts towards left. Here we have plotted the critical temperature as a function of strength of random field for 2 different strength of anisotropy. You can see that the critical temperature decreases monotonically if we increase the strength of random field. So the random field introduces a quench random disorder to the system and that reduces the TC. Certainly the upper points corresponds to the higher value of the anisotropy. That is, the anisotropy increases the critical temperature by disrupting the SO2 symmetry of the system. So the anisotropy disrupts the SO2 symmetry of the system and the critical temperature increases with the increasing value of anisotropy. On the other hand, the random field disrupts the spin orientations and reduces the critical temperature by introducing disorder into the system. So the interplay between constant anisotropy and random field offer a fascinating competition. By tuning these two factors very carefully, we can measure the critical temperature for an isotropic system, that is 2.206 for a 3D XY ferromagnet. The strength of random field that is required to restore the isotropic XY ferromagnet critical temperature is defined as the compensating field. We have measured the critical temperature for some values of random field for different strength of anisotropy. By interpolating them, we can get the critical temperature of the isotropic XY ferromagnet, that is 2.206. Here we have plotted the compensating field as a function of strength of anisotropy. You can see that the compensating field increases linearly with the increase of anisotropy strength. This line corresponds to the isotropic critical temperature, that is 2.206. Below this we get the ferromagnetically ordered phase, and the above part is the disordered phase. So we have seen that the random field with full circular symmetry can reduce the critical temperature and also give rise the long range ferromagnetic order in the XY system, even in 3 dimensions. So what happens if we circumscribe the angular field by an angular window? This type of field shown in this figure, where 2 of delta phi, that is phi 1 minus phi 2, define the angle of the field width actually. So these are the numerical results for random field with conical symmetry. If the similar kind of results we have observed in this case also. If we cool the system gradually, the magnetization grows and susceptibility shows a peak. Also the corresponding specific heat get peak near the transition temperature. Here you can see that the critical temperature also decreases if we increase the angle window. The critical, here we have plotted the critical temperature as a function of angle width. If we start to constrict the angle width from the circular symmetry, that is 2 pi, to angular window, the critical temperature increases monotonically. This figure shows the critical temperature as a function of angle window for different field amplitude for a constant value of anisotropy. So random field distributed in a conic also leads to the order disorder transitions. Any phase transition should be formalized by finite size scaling analysis. So we have also done the finite size scaling analysis for both of these cases. The upper part shows the first type of random field, that is the random field with full circular symmetry. The inset figure shows the thermal variation of susceptibility for different system sizes. If we increase the system sizes, the susceptibility peaks, susceptibility maxima increases. That is therefore the phase transition is supported by growth of
2 more minutes.
Critical correlations, that the susceptibility shows a divergence property at the vicinity of transition temperature. From the maxima point, we have plotted the plotted against the system size in a log scale. We can get the critical exponent, assuming the critical exponent as susceptibility goes as L to the power gamma by nu and specific heat goes as L to the power alpha by nu. We can estimate the critical exponent for this type of phase transition. Let us summarize all the results. First, we have shown that there is also long-range ferromagnetic order exists in case of random field XY model, even in 3 dimension, in the presence of constant anisotropy. Actually, constant anisotropy breaks the inherent SO2 symmetry of the model. That that's give rise to the critical that gives rise to the conventional phase transition in presence of random field. The directional preference imposed by the constant anisotropy can also be cranked down by applying the random field and the compensating field, that is the prescribed amount of field which retains the critical temperature for an isotropic XY ferromagnet in case of 3D was found to depend linearly on the strength of anisotropy. These are the references I have talked about. This work, this has been recently accepted on JMM, and I would like to thank my supervisor, Professor Mukti Acharya, for his guidance and invaluable advice. And my research fellowship is funded by UGC. And thank you.
So now we are open to a few questions. And we will be taking a few questions only because we are running out of time.
So, Anita, can you tell me what is the origin of anisotropy? What is the origin of anisotropy?
Crystal structure, structural.
Certain crystal structure may have anisotropy, but what is the actual physical origin of this anisotropy appearing in crystal material? All the materials will not show this anisotropy. Other materials will show, but what is the origin?
Electronic structure of the atom.
But I want to know what electronic structure, and that is also another question is that the value of J for this kind of ferromagnetic materials is positive, negative, or zero?
Negative.
For ferromagnetic.
J for ferromagnetic, J is greater than 0 to
Almost the last part you figured it with some exponent.
Yes.
And it's I mean, how do you know is gamma by mu? You don't you do not fit them separately, gamma or alpha, right? You're not able to determine. You can only just say that L is to the power of something, and whether it is alpha by mu or gamma by mu is. Are you assuming something already?
Please repeat the question.
So I'm saying that this fitting is L to the power of some exponent, but that exponent could be anything, right? I mean, how do I know that it is the ratio of these two factors? Because it's a number. Yeah, I mean that's what the question is, whether we can define gamma by mu or is it just a sub exponent? Separate, I mean, gamma probably has some meaning, or mu would have some meaning, whether there is any meaning here. Separately, do you know gamma or alpha? Can you determine them?
I have not determined.
Last question.
BKT transition in case of 2D XY ferromagnet, the unbinding of vortices give rise to the phase transition without having any long range order. Any system which can be modeled as R e to the power I theta, so you can do that for this type of material.
What is the full form of this BKT?
I don't know. I am not sure about the B, but K. Costerlis and Thaulis. That was the Nobel Prize of 2016.
Thank you.
So now we call the last but not the least speaker for this session, Dr. Ramu Kumar Yadav. So he has done his PhD under Dr. Rajiv Kapri on non-equilibrium soft condensed matter physics. He's currently working as a postdoctoral research associate at the Department of Physics of Complex Systems, SN Bose National Center for Basic Sciences. So over to you, Dr. Yadav.
Good afternoon to all of you. I shall be talking about the study of unzipping transition in an adsorbed polymer by a periodic force. So what happens, there are many cases where we see that the polymers gets adsorbed on some attractive surface. So now we need to remove that polymer from those surfaces. So we can remove the polymer from the attractive surface in two ways. One, with respect to temperature. Suppose if I am changing the temperature of the system, then we see that at a specific temperature, the polymer can be removed from the attractive surface. The second is that we can do it with respect to the force. If at a particular temperature, which is less than the desorption temperature of the polymer, if we keep increasing the force, then we see that at a specific force, the polymer get removed from the surface, and we call that force a critical force, and that critical force GC is a function of the temperature. This study is very important because it finds many applications in the like the process lubrication, adhesion, surface cohesion, and many other biological process. So what happens when I study this system with respect to the force, then there are two cases of the force. So in one case, I can apply the static force and study the system. That I am not going to present here. But in static force case, and the second one is the periodic force case, the dynamic force case. In dynamic force, which I will present the result for the dynamic force, we see that for the dynamic force cases, the analytical and exact solutions are not possible. So we use the Monte Carlo simulation to study the dynamic force case. And we see that there is a hysteresis between the force and distance isotherms. Here is the model of our system. So in the model, we model the polymer by a self-avoiding random walks. And self-avoiding random walks, and we see that the walk starts from here 0, and it directed toward the z directions and self-avoidance. This direction nature takes care of the self-avoidance. And the second thing that in this model, if this monomer is, and we also put a wall, an attractive wall at x equal to 0. This is the n wall attractive. Whenever there a monomer of the polymer is in contact with the wall, our system gets the energy minus epsilon, and we can see minus epsilon. And in our study, what we do, we keep this end of the polymer fixed, and every time we apply a force here, you can see that GT, periodic force, and pull the polymer away from the wall, GT. And our response in our study, the response is the distance of this last monomer from the wall. And we have considered the three different kinds of the wall. So this one is the soft wall. We can call this one is soft wall case, because in this case polymer can explore the both region of the wall and the region have the same affinity for the wall. In this case, we call the wall separating two different kinds of the media, because in these two different kinds, here the polymer will have a different kind of affinity, here the polymer will have a different another affinity. And the last one we call the hard wall case. In this case, polymer is not allowed to go the region x is equal to 0. It can be adsorbed here and go in the x is greater than 0 region. So what we see, force, the nature of in the force, we consider the force. This is not working. So the force we apply, this is the GT is equal to G naught sine omega t. G naught is equal to the amplitude of the force, and we use the amplitude greater than the critical force so that it will unzip them. And omega is the frequency of the force. And we perform the Monte Carlo simulation of the model. So here we plot. Here you can see that the x-axis is the temperature. The green color is the force, and these dotted lines are the response x. And I have scaled that x as a length of the DNA. So we see the first column is at a higher frequency. The second column plots are a lower frequency. You can see that for the higher frequency, the extensions are very smaller. For all the three cases, this is the soft wall, this is the two different media, and this one is the hard wall. You can see the extension is very small in all cases. This is because at the higher frequency, the force system does not get enough time to relax and the response is very smaller. And one more point here, we notice that there is always a phase lag between this applied force and the response of the system. The lag is very finite at the higher frequency. However, the lower frequency, where now the system has enough time to relax, we see that the extension is comparatively very high, and also we see that this phase lag decreases. You can see that the maxima of both is coinciding. So because of this phase lag, when we take more than 10,000, we can say that significant number of cycles, and we average those cycles, we find closed loops. You can see that we find closed loops. This is the average axis, and this is the x-axis is the force. In all cases, we find the hysteresis loops. So this now I have considered the first column is a force amplitude which is very close to the phase boundary. The last column is a force amplitude which is far away from the phase boundary. You can say that, and the first row is the soft wall case. We see that for the soft wall case, we get the hysteresis loops which are symmetric in the both direction. And at the higher force amplitude, you see that the only difference is the loops gets the higher in the size. In the two different media case, we see that when the force amplitude is very small, you see that when the force amplitude is 1.0. So in this case, the loop we get the loops only in the x is greater than 0 region, but not in this direction. However, when I increase the force amplitude for the wall separating two different media, we see that now the polymer gets enough, and now polymer gets the potential, which is the finite potential in the x is less than 0 region, and able to explore the more region. And that is because we get the loops in this region also. And in the hard wall case, we can see that this hard wall case, the loops are very similar in both the force amplitude cases, and the loops are only in the positive direction. X is greater than 0 region. So what we see that we calculate the area of these loops. We use some library and calculate the area of these loops, and I plot the area. So this one is for the soft wall case, and for the two force amplitude, this is the lower force amplitude, this is the higher force amplitude. You see that when I increase the frequency, the area of the loop increases, and at a specific frequency, the area of the loop gets the maximum value, and we call that specific frequency as the resonance frequency. And as I further increase the frequency, the area gets decreases, and it tends to 0 at the very lower frequency, because that is the case of the static force case, convergent to static force case. So what we say that in soft wall case, for the both amplitude we get the similar behavior. One more point which we notice from here, that as the length of the polymer increases, we see that this maximum value of area increases. And also we notice from here that as the length increases, this resonance frequency for this, you can see the resonance frequency is here, but as the length increases, the resonance frequency shifts towards the lower direction. So this suggests us a scaling of this form. And when we use this scaling of this form, and for the values of delta is equal to 1 or z is equal to 1, we get a very nice collapse of data for both of the cases. Now this is the case of the wall separating two different kind of media. So as we can see that here, the difference only is at the lower force amplitude, a system does not overcome the potential in the negative direction. So we see that only one peak we get. However, we see that because of this, we get the two loops at the two peaks in the wall separating two different kind of media. And you also see that are the same kind of scaling. This also shows a very good nice collapse of data in this case too. And this one is the last case, hard wall case. So what in hard wall case we see that the behavior at the smaller force amplitude is similar to the previous cases. However, in this case we see that when we increase the force amplitude, the system shows the similar behavior here, but it show an oscillatory. These are the secondary peaks, which are in the higher frequency region, and these are the secondary peaks. And these secondary peaks numbers increases as will increase the length of the polymer. And this also show a very nice collapse of the data or the same critical exponents. And now we talk about the scaling, low frequency scaling. So we see that in this case, we see that this. Yeah, I am talking about this part of the low frequency part of the system. So we see that for the three different force cases, one particular length of the polymer, we get a very nice collapse of data with this function. X-axis is equal to omega to the power beta and G naught minus omega c. Alpha and value of alpha and beta are alpha value is equal to 1.0 plus minus 0.5 and beta value is equal to 1.25. We get the for three different lengths and one kind of wall, and this is the second kind of wall, the two wall separating two different media, and this one is the last one, hard wall case. And the same, they are so nice data collapse for the same values of the critical exponent. And now I summarize my presentation. So we observed that the force distance isotherm show the hysteresis loops in all the three cases. We found that the A loops show the monotonic behavior as frequency is varied, keeping the amplitude constant. And we also see it showed peaks of different heights for the wall separating two different kinds of media. And we also noticed that there are secondary peaks in for the hard wall case. And the last we find the A scaling, which is the very robust scaling and exactly similar to some other Monte Carlo simulation study, and the values of scaling are there.
So now we are open to a few quick questions. So any questions from the audience?
Small question. Can you repeat the motivation of this problem? You said something in the beginning, but
so suppose sometimes polymers gets agar. Suppose there are two kinds of liquid and they have immiscible liquids, but they cannot mix. So there are some cases when the polymer gets agar at that boundary interface. We need to remove those things. And those are the motivation basically. And there is a biological relevant process, DNA, unzipping of DNA. Similarly, I have studied similar model of a DNA. So it also motivated like that. Periodic force. Static force and then periodic force.
Which is the forcing across omega squared t. So what will happen to these peaks?
What I have amplitude and frequency in force. So that is the result I saw. The variation of frequency and all those results are talking about the. I plotted all results with respect to frequency.
Last question.
Go to the fifth slide.
This?
What is this Metropolis algorithm actually?
Metropolis algorithm.
What is this Metropolis algorithm?
This is basically we generate a difference. Suppose polymer is in a state. Suppose it is completely adsorbed on the wall. Now I want to generate the different state. So now I randomly remove one monomer and then calculate the whole energy of the system next time. And then I will say system will go in that state or not. That will define by the Metropolis algorithm. If energy is less than to the previous case, I will let my system go there. If the energy is greater than the previous case, then I will use the Boltzmann factor. You have no e to the power minus delta E upon kt. Then I will let the system go there. That.
Can I go to the ninth slide? Ninth. Just I want to get your top right diagram. You can see the 128 line, and the second peak you can see there is a the fitting is like a sharp fitting is there. The point is just like that, and like it's like a pyramid being formed at the right hand side peak. Can you tell me what is the fitting behind that logic?
No, I am not getting your point. What are you saying? You are talking about this plot.
120, go to the 120.
If there is a sharp,
Go to the
This.
120 that, and the sharp one. The next peak. Not that one.
This.
Next peak. Yes. This. Why this kind of pyramid thing is there?
No, I need more data point. No, I will. No.
We are given them no, this is not fitting.
No.
even the red one is wrong.
Yes, actually that's why. Just joining the data. Okay.
Okay.
Thank you, Dr. Yadav. And thank you everyone for joining with us for this session. So thus we conclude our first oral presentation session for the day. So I'm handing over the mic back to Shagnik. So Shagnik, please take over.
Thank you, Pomannu. So now it's time for the lunch break. So all of you are requested to take your food packet from outside. So in the meantime, there will be a poster presentation session. So all the participants are requested to take their positions in front of your posters. And we will meet here again at 2:30 p.m. for the next session.